Climate change is already causing the Earth to warm at an alarming rate, with current projections suggesting it will be up to 4 degrees Celsius warmer by the end of the century. Organisms have evolved to live in particular environmental conditions, so this rapid warming may be beyond their adapted capacities, potentially leading to catastrophic consequences for species' performance and survival. In order to predict how plants and animals will respond to predicted future environmental conditions, we need to understand their potential to acclimate and adapt. In the marine environment, scientists have generally collected animals from the wild and exposed them for a relatively short time to the warm conditions predicted in the future, from days to weeks or months. When this type of experiment is done, most species, including fish, suffer declines in aerobic capacity, reproduction, growth, and sometimes they aren't able to survive. But climate change isn't going to happen that quickly. For most species, it'll happen over many years and generations. Our research studies how a coral reef damselfish could cope with the predicted ocean warming over multiple generations. Since we already know that warming over short timescales produces negative effects to aerobic performance, we set out to determine what would happen if fish were allowed to fully develop in warmer conditions from early life compared to present-day temperatures. We found some slight improvements to performance, but the fish were still not performing optimally. Next, we wanted to investigate how a second generation performs in warmer water. So the fish that developed in warmer temperatures for their entire life then had offspring, which we again reared in the same warm conditions as their parents. This second generation fully restored their aerobic performance back to current day levels. So these damselfish can acclimate over only a couple generations, but how were they able to acclimate so quickly? To do this, we needed to take a closer look at the molecular level, take a snapshot of all the genes that were activated or inactivated to allow these fish to cope in warmer water. So which genes play a critical role in thermal acclimation? To work this out, we first identified the genes that were significantly different among treatments, and then compared the gene activity levels with aerobic performance. This allowed us to identify 53 key genes. These genes were primarily involved in metabolism, but also immune, stress, and growth-related processes. Surprisingly, none of the most common genes assessed in thermal stress studies, the heat shock protein genes, were activated in our multi-generational acclimation study. Interestingly, the same suite of genes and processes were activated in both generations, just to a greater extent in the second. This, in combination with the rapid acclimation, suggests an exciting mechanism called epigenetic inheritance might be at play, where the parents prime their offspring for survival without actually changing their genetic code. An exciting prospect we're researching in more detail now. By identifying the genes involved in thermal acclimation of key performance indicators like aerobic capacity, we have gained an important understanding of the cellular processes that marine animals can use to adjust to future climate change. Our aim is to now use these genes in other populations and species to determine the animals that are most at risk in the years to come.